Endocrinology Introduction. Lecture 3. Hormonal actions mediated by intracellular receptors. The cholesterol derivatives, steroid hormones, are lipid-soluble, and are thought to enter cells by diffusion through the lipid bilayer of the plasma membrane. The thyroid hormones have large nonpolar constituents. They may penetrate cell membranes by diffusion, but carrier-mediated transport appears to be the primary means of entry. These hormones bind to receptors that are located in the cell nucleus or cytoplasm and produce most, but not all, of their effects by altering rates of gene expression. Steroid hormones bound receptors. Receptors bound to steroid hormones bind to specific nucleotide sequences in DNA, called hormone response elements, HREs, located upstream of the transcription start sites of the genes they regulate. The end result of stimulation with these hormones is a change in genomic readout, which may be expressed in the formation of new proteins or modification of the rates of synthesis of proteins already in production. General model of steroid hormone action. Steroid hormones penetrate the plasma membrane and bind to intracellular receptors in the nucleus or cytoplasm. Binding activates the receptor, which forms complexes with other proteins and binds to specific acceptor sites, HRE, on DNA. This initiates transcription and formation of the proteins that express the hormonal response. The steroid hormone then is cleared from the cell. Activation of steroid hormone receptors. Inactive receptors associated with other proteins react with hormone, shed their associated proteins, and change their conformation. They can then form dimers that bind DNA, and a variety of nuclear peptide regulators of gene transcription. Receptors for thyroid hormone. They belong to another closely related group within the same family of proteins as the steroid hormone receptors. Unlike the steroid hormone receptors, these receptors are bound to their HREs and DNA even in the absence of hormone. In the absence of hormone, these DNA-bound receptors form complexes with other nuclear proteins that may promote or inhibit transcription. Upon binding its hormone, the receptor undergoes a conformational change that displaces the associated proteins and allows others to bind, with the result that transcription is either activated or suppressed. Target cell response. Many steps lie between activation of transcription and changes in cellular behavior. These reactions necessarily occur sequentially and each takes time, and they include synthesis and processing of RNA. Transcription, 40 nucleotides/S, 10,000 nucleotides takes almost 5 minutes. RNA processing takes 20 minutes. Exporting it to cytosolic sites of protein synthesis. Protein synthesis itself. 15 amino acids per second. Protein processing. And delivery of the newly formed proteins to appropriate loci within the cells. Termination of signal. As hormone blood levels decline, intracellular concentrations also decline. Because binding is reversible, hormones dissociate from receptors and are cleared from the cell by diffusion into the extracellular fluid, usually after metabolic conversion to an inactive form. Unloaded steroid receptors dissociate from their DNA binding sites and regulatory proteins, and either recycle into new complexes with HSP90 and other proteins, or are degraded and replaced by new synthesis. Termination of signal. RNA transcripts of hormone-sensitive genes are degraded usually within minutes to hours of their formation. The proteins are degraded with half-lives that may range from seconds to days. Thus, just as there is delay in onset, effects of the hormones that act through nuclear receptors may persist after the hormone has been cleared from the cell. Hormonal actions mediated by surface receptors. The protein and peptide hormones and the amine derivatives of tyrosine cannot readily diffuse across the plasma membranes of their target cells. These hormones produce their effects by binding to receptors on the cell surface, and rely on molecules on the cytosolic side of the membrane to convey the signal to the appropriate intracellular effector sites that bring about the hormonal response. Hormonal actions mediated by surface receptors. The G-protein coupled receptors. Receptors that signal through tyrosine kinase. The G-protein coupled receptors. The most frequently encountered cell surface receptors belong to a very large superfamily of proteins that couple with guanosine nucleotide binding proteins, G proteins, to communicate with intracellular effector molecules. G-protein coupled receptors are crucial for sensing signals in the external environment such as light, taste, odor. Signals transmitted by hormones. More than 1,000 different G-protein coupled receptors are expressed. Activation of G-protein coupled receptor. Resting state. Hormone binding produces a conformational change in the receptor. This causes the alpha subunit to exchange GDP for GTP, dissociate from the beta, gamma subunit and interact with its effector molecule. The beta, gamma subunit also interacts with its effector molecule. The alpha subunit converts GTP to GDP, which allows it to reassociate with the beta, gamma subunit, and the hormone dissociates from the receptor, restoring the resting state. Termination of signal. Dissociation of hormone from its receptor. G-protein coupled receptors may be inactivated by phosphorylation, the second messenger concept. For a hormonal signal that is received at the cell surface to be effective, it must be transmitted to the intracellular organelles and enzymes that produce the cellular response. To reach intracellular effectors, the G-protein coupled receptors rely on intermediate molecules called second messengers, which are formed and/or released into the cytosol in response to hormonal stimulation. A single hormone molecule interacting with a single receptor may result in the formation of tens or hundreds of second messenger molecules, each of which might activate an enzyme that in turn catalyzes formation of hundreds of thousands of molecules, a product. Second messengers can also promote the phosphorylation of transcription factors, and thus regulate transcription of specific genes in much the same way as discussed for the nuclear receptors. The second messenger concept. The cyclic AMP system. The calcium commodulin system. The DAG and IP3 system. DAG, diacylglycerol. IP3, inositol trisphosphate.